Good morning. It's September 2nd, and this is your daily brief in science. Here's everything you need to know. A groundbreaking development in chocolate production is taking shape in California. California Cultured, a plant cell culture company based in West Sacramento, is pioneering a method to grow cocoa from cell cultures, significantly reducing the maturation time from the traditional 6 to 8 months to just 1 week. The company plans to launch its lab-grown cocoa products by next year, aiming to address the increasing global demand for chocolate. To facilitate this, California Cultured is seeking FDA approval to label its lab-grown cocoa as chocolate, highlighting its genetic similarity to traditional cocoa. The U.S. chocolate market surpassed $25 billion in sales in 2023, with demand projected to outpace cocoa supply due to environmental challenges and increased consumption. Earlier this year, a surge in cocoa prices was driven by high demand and crop issues in West Africa, which produces most of the world's cocoa. Climate change poses a significant threat to cocoa production in rainforests, prompting research into alternative growing methods and substitutes. Notably, Mars is investing in research at the University of California, Davis, to enhance cocoa plant resilience against disease and support farmers. In addition, Celeste Bio in Israel is cultivating cocoa bean cells indoors to produce cocoa products that are less affected by climate change. This trend indicates a shift towards innovative solutions in cocoa cultivation and production to meet the growing demand for chocolate. Despite most cocoa being grown in Africa, only a small percentage is consumed there, underscoring the global nature of chocolate demand. Consumer interest in sustainable and ethical food sources, particularly concerning child labor in cocoa farming, is also driving innovation in the industry. Alongside these developments, Planet A Foods is working on a chocolate alternative called Choviva, made from oats and sunflower seeds, aiming for mass-market appeal without replacing high-quality chocolate. Recent advancements in data stream classification techniques are making waves in the renewable energy sector, particularly in wind and photovoltaic power generation. These innovations are crucial as they address the challenges posed by data drift, which refers to the changes in data distribution over time that can significantly impact machine learning models. Fault diagnosis plays a vital role in photovoltaic power plants, which are particularly susceptible to complex environmental conditions. To enhance fault diagnosis and maintenance strategies in both wind and photovoltaic systems, comprehensive monitoring solutions that analyze component-level data are essential. This focus on detailed data analysis is increasingly important as the operational efficiency of power generation systems relies on effective data management. The authors of a recent study emphasize the importance of selecting computationally efficient outlier detection methods tailored for specific applications. They provide guidelines for choosing appropriate methods based on detection challenges and the characteristics of the data. One effective tool highlighted is the exponentially weighted moving average control chart, which can detect abnormal states such as open and short circuits, pinpointing the specific string where a fault occurs. The rapid growth of data generated from clean energy sources like wind and solar power necessitates the development of efficient methods to extract valuable insights from these data streams. Wind turbine generators, often facing downtimes due to manufacturing errors and harsh conditions, underscore the need for better operational strategies. Existing fault diagnosis methods leverage both electrical indicators and vibration signals, showcasing the role of machine learning in enhancing accuracy and reliability in energy production. A recent study has flipped our understanding of butterfly wing patterns on its head. Led by Luca Livraghi from George Washington University and the University of Cambridge, this groundbreaking research has identified an RNA molecule as the key determinant of black pigment distribution on butterfly wings, challenging the long-held belief that proteins were the primary influencers. Previously, it was assumed that protein coding genes were essential for generating pigments. However, this study highlights a specific RNA gene that controls the development of dark pigments during the metamorphosis of butterflies. The researchers observed that the expression of this RNA was closely linked to the formation of black scales during wing development. Described by researcher Arnaud Martin as an evolutionary paintbrush, this RNA's role was examined across various butterfly species that diverged approximately 80 million years ago. The findings revealed that this RNA is crucial in controlling the placement of dark pigments on the wings. Using CRISPR genome editing techniques, the team demonstrated that the absence of the RNA-producing gene led to butterflies losing their black pigmented scales. Published on August 30, 2024, in the Proceedings of the National Academy of Sciences, this study emphasizes the importance of recognizing overlooked genetic mechanisms. The implications of this research extend beyond butterflies, as it challenges existing beliefs about genetic regulation and opens avenues for future studies on the evolution of visible traits in animals. Joe Hanley pointed out the repeated significance of this RNA across multiple butterfly species, suggesting that there may be other overlooked phenomena in genetic research. Ricardo Papa added that this RNA gene represents an ancestral mechanism for wing pattern diversity, rather than a recent development, further emphasizing its importance in the genetic orchestration of color arrangement in butterfly wings. The latest findings from the ABYSS trial, presented at the ESC Congress 2024, raise critical questions about the management of beta-blocker therapy in patients with a history of myocardial infarction. This trial involved 3,698 patients who were randomly assigned to either continue or interrupt their beta-blocker treatment, with a follow-up period of at least 1 year. All participants had a left ventricular ejection fraction of at least 40% and had not experienced any cardiovascular events in the previous 6 months. The primary outcome, which included death, non-fatal myocardial infarction, non-fatal stroke, or hospitalization for cardiovascular reasons, occurred in 23.8% of the interruption group compared to 21.1% in those who continued their medication. This resulted in a risk difference of 2.8 percentage points and a hazard ratio of 1.16, indicating that non-inferiority was not achieved. Notably, patients who stopped taking beta-blockers experienced higher rates of cardiovascular hospitalization, with 18.9% in the interruption group versus 16.6% in the continuation group. Death rates were similar between both groups, at 4.1% for the interruption group and 4.0% for the continuation group. Myocardial infarction rates also remained comparable. Furthermore, discontinuation of beta-blockers was linked to increases in blood pressure and heart rate after 6 months, suggesting negative cardiovascular effects. Contrary to expectations, there was no improvement in quality of life for patients who interrupted their beta-blocker therapy, as measured by the European Quality of Life 5 Dimensions questionnaire. Professor Johanna Sylvain, the trial's lead investigator, emphasized that these findings do not support the interruption of chronic beta-blocker treatment in post-M1 patients. Ongoing trials aim to provide additional data on the safety and efficacy of discontinuing beta-blocker therapy in this population. These results challenge existing guidelines that suggest beta-blockers can be safely discontinued after 1 year in certain patient populations. Morphine is a powerful opioid painkiller known for its ability to block pain signals while enhancing pleasure. However, its use comes with significant risks, including addiction and overdose, which complicate its role in healthcare. Recent research has turned its focus on the specific neurons responsible for morphine's pain-relieving effects, particularly those located in the rostral ventromedial medulla, or RVM. These neurons are critical for pain inhibition as they connect to the spinal cord. Studies have shown that inactivating this group of neurons can completely eliminate the pain-relieving effects of morphine. On the other hand, reactivating these neurons has been found to restore the pain relief that morphine typically provides. Patrick Ernfors, the lead researcher of the study, emphasizes the importance of understanding these neural pathways to mitigate the dangerous side effects associated with morphine. The goal of this research is to uncover the mechanisms behind morphine's effects, paving the way for safer pain management strategies that could lower the risks of addiction and overdose. This significant study received funding from notable organizations, including the Swedish Research Council, the European Research Council, and the Knut and Alice Wallenberg Foundation. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.